Good afternoon. On behalf of the Biochemical Society and the Portland Press, I am pleased to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. My name is Lukas Tamayo. I am a postdoc in the group of Andrew Jackson at the University of Edinburgh, and I am interested in DNA replication defects. In this webinar, we will hear from Dr. Rashmi Panigrahi from the Memorial University in Newfoundland in Canada. Changes in chromatin, the functional state of DNA in a cell, is integral to the DNA damage response. In her talk today, Rashmi will discuss the use of biochemical tools to obtain structural insights into phosphorylated nucleosome in the presence and absence of BRCT domain containing repair factors. Questions will be asked after Rashmi's presentation, but you can send in your questions during the talk. If you have a question, please type in the question in the question box as shown in the image on the screen, and we will try to answer as many as time allows. So we welcome Rashmi to her talk.
Thank you, everybody, and thank you, Lucas, for chairing the session and inviting me. Thank you, Lucy, for organizing the event, and I'm really honored to be speaking today before all of you in this biochemistry focused webinar series. And today I'm going to talk a story about a project that I did in my postdoc in Professor Mark Glover's lab, where we integrated structural biology and biochemical tools to uncover nucleosome dynamics. So Professor Mark Glover's laboratory has been focusing on gene regulation aspect, both from prokaryotes and eukaryotes. Crystallography is the primary technique in our lab, and these are some of the high resolution, beautiful structures, particularly from the eukaryotic arm. The commonality between all these proteins is that they belong to the DNA repair pathway, and especially of DNA double-stranded break repair pathway. And as we know, there are two main kinds of this repair. One is the non-homologous end joining and the homologous recombination. When I joined the lab, Dr. Glover was actually interested in understanding how these previously solved repair proteins interact with the chromatin. Now, about the chromatin, and this is a very general overview. The chromatin can form a open conformation, which is the euchromatin, or a very condensed conformation, which is called heterochromatin. And the basic unit of the chromatin is the nucleosomes. So the nucleosome, as shown here, consists of a proteinaceous core, which is made up of these proteins called histone proteins, and there are 8 of these proteins belonging to 4 classes, which is H2A, H2B, histone H3, and H4. And then the core protein complex is surrounded by these DNA double-stranded thread kind of structure, which wraps around probably 1.6 times. And in the entry-exit site of the DNA, as you can see here, there is a 5th histone protein that can exist, which is the H1 histone, which locks the nucleosome in a closed conformation so that the histone core proteins do not come out of the nucleosome. Now, these histone tails, which are very flexible, are the sites of post-translational modifications. And these post-translational modifications are the signals, here we call it as the PTM, and this signal different processes as shown here. And today, the main focus is DNA repair, and particularly DNA damage signaling of the DNA repair pathway. Now, the histone post-translational modifications are installed, read, and removed by different proteins. So they are installed by this class of enzymes called writers, and some of the examples are shown here. Now, once the post-translational modification is installed, they are read by specific domains of proteins called as readers. And the protein of interest today are these reader class of proteins, which has a specific domain, which is called the BRCT repeats. Now, once the cellular process is over, triggered by the specific post-translational modification, these modifications are removed by a class of enzymes called as erasers, and some of the examples of those classes are written here. So what happens during DNA double-strand break? The break is recognized by sensor protein complexes, which then recruit mediator proteins, which gets phosphorylated, and thus the mediator is activated. And then the activated mediator then phosphorylates the nucleosomes, which contain the H2AX variant containing nucleosome in the periphery around megabases of this damaged site. And this is called DNA damage signaling. And this signal, this phosphorylation of H2AX, which is otherwise called gamma H2AX, spreads around megabases around this DNA damaged site. And this phosphorylation is kind of recognized by the reader proteins. And today we will talk about two proteins, which is microcephalin protein 1, MCPH1, and the mediator of DNA damage checkpoint protein 1, MDC1. And these proteins recognize this phosphorylation via their C-terminal BRCT domains. And this recognition leads to the recruitment of downstream repair factors and the ubiquitinating machinery, etc. Now, at a more molecular level, this is the C-terminal of the H2AX histone, wherein the serine 139 is the site that gets phosphorylated to form the gamma H2AX. And so in healthy cells, the terminal tyrosine of the H2AX variant is basally phosphorylated by a particular kinase. Now, upon DNA damage, the serine 139 gets phosphorylated. So you can find a doubly phosphorylated form, which is recognized by the C-terminal BRCT of MCPH1. Now, this terminal phosphorylation is removed by a phosphatase, and so we are left with the serine phosphorylation, which is still can be recognized by the MCPH1 BRCT domain, and eventually the MDC1 comes in and recognizes this serine phosphorylation via its BRCT domain. So taken together, for many decades, it has been known that BRCT domains are the key readers for DNA damage signaling. Now, this is the domain organization of these two proteins, and there is a lot of disorder, as you can see by this straight line. But most importantly for the talk, we focus on the BRCT repeats, which are present in the C-terminal of both the proteins. Now, interestingly, the structures of these domains are solved. Sequence-wise, they are quite different, but structure-wise, they are very similar, as you can see here. And the structures are solved by our lab and others. So the structures are color-coded over here. Pink throughout the slide would be for MCPH1 BRCT, and blue throughout the slide is for MDC1 BRCT. Now you can see the gamma H2AX phosphopeptide. Here is the sequence where the serine 139 is phosphorylated, was co-crystallized. The protein peptide complex is shown here. And what you can see in both the cases, that is MCPH1 and MDC1, the phosphoserine is buried in a pocket in the BRCT1, which is the N-terminal BRCT repeat. And between the BRCT1 and BRCT2, there is a hydrophobic patch where the terminal tyrosine is buried. Now, this has been evidenced multiple times by multiple groups, including ours. Now, the question we asked in this project is that how does the BRCT interact with not the gamma H2X peptide, but more in the nucleosome context? Particularly, does the binding of BRCT to this gamma H2X nucleosome lead to any structural rearrangement, either in the histone or in DNA or both? So to address this question, we first prepared phosphorylated nucleosome, and then we formed the complex of this gamma H2X nucleosome with BRCT, and then we performed structure solution studies using cryo-electron microscopy and MD simulations. So first, in order to prepare the gamma H2X containing nucleosomes, containing octamer, first of all, we did something as a trick using cloning method. So we truncated the C-terminal region of H2AX, these residues, this peptide, and here we have the serine 139, and replaced it with the intine sequence followed by the chitin binding domain. Now, intine is a self-splicing protein, and finally what we get is the whole octamer with the truncated tail containing the intine chitin binding sequence. And then the intine thiolysis is started by addition of a thiol containing compound, which is called mesna. And that leads to a formation of a thioester containing tail. And then the native chemical ligation is initiated by addition of the synthetically procured peptide, which has this phosphoserine in it, and TSEP. And after the ligation reaction, you can see the phosphoserine is nicely incorporated to the H2AX tail, and now we call this as the gamma H2AX octamer. We incubate it with 145 base pairs double-strand DNA and perform salt gradient dialysis to remove the salt. And then we notice it on we visualize it on the native page. And so when we have incubation of a fixed concentration of DNA and increasing concentration of histone octamer, what we see at one point of time, we have fully formed nucleosomes. That means we have no unbound DNA. And so there we have, and we can use it for further binding and structural studies. Now, for one of the binding studies we performed in this project was microscale thermophoresis, where we purified the BRCTs and then labeled the C-terminal histidine tag of the BRCTs with show that BRCT actually specifically interacts with the phosphorylated nucleosome, phosphorylation being on the serine 139 on the H2AX C-terminal tail. Now, for the structural studies, we performed cryo-electron microscopy, wherein we had purified samples of nucleosomes, gamma H2AX containing nucleosomes, or BRCT bound to gamma H2AX containing nucleosomes. And the steps of cryo-EM are pretty standard, but for the general audience, very quickly, the sample is applied to the grids. And here you can see a non-aggregated homogeneous sample, and the excess liquid of the sample is blotted away. So you have a thin film of sample in the grid, and then the grid is plunge frozen in liquid ethane, and you have a nice layer of your particles in vitreous ice, and then you load the grid on the transmission electron microscope, and when the electron beams hit the grid, we get these images which are collected on the detector. So moving on, the first view of our grids are shown here. This is for the H2AX NCP, and this is the gamma H2AX NCP. Now, at first we see that both of the grids look quite similar. In other words, we have top view of nucleosomes that are pointed using arrows, And then there are side view of nucleosomes, which are arranged in a stack order, as you can see in both the grids. When we performed 2D classification, we found that we got beautiful mononucleosomes as well as stacked nucleosomes. Now, the overlay of the model of the canonical form of the phosphorylated and non-phosphorylated nucleosomes show that they were almost identical. The histone tails were missing, and that is quite expected, shown by others as well, because of the flexibility issue. And as you can see here, all the histone proteins are color-coded. Now, in total, we solved 9 different structures. 5 of them were different versions of mononucleosomes, and 4 of them were different versions of stacked nucleosomes. And the highlight of this work is the stacked nucleosomes, and that has never been reported before in the way we have done the experiments. So in the essence of time, I will only go over 2 kinds of stacked nucleosomes out of 4. One of them here is the H2AX containing nucleosome stacks. And here we have our nomenclature, where the bottom nucleosome will be henceforth referred as NCP1. The top nucleosome will be referred to as NCP2. So in this nucleosome stack, as you can see, NCP2 has kind of moved towards, or slided, can I say, towards the right of the NCP1. So we termed it as right-hand offset stack. Now the interesting part is the interface, which has not been reported previously. So there are 3 kinds of interface, if you focus on this here. So we have a histone DNA contact, and the histone DNA contact is made out of H4 histone and the DNA of NCP2. And then the 2 major contacts over here in the center are is made by the H2B elements of NCP2 and H2B elements of NCP1. And again, all the elements are color coded here. As you can see, it is H2B. Now, think of this nucleosome interaction as a sandwich, and if we open it up to have a better view of the interaction, we can see that here again color-coded H4 N-terminal arginine 24 interacts with the DNA of the NCP2, and similarly the H2B elements of NCP1 interacts with similar H2B elements of NCP2. Now, the other form of nucleosome I have picked is from the gamma H2AX nucleosome, and these we call it as parallel stacks. And this is a video. Here we have the cryo-EM density, as you can see, and the model will be shown in a few seconds. That's here, and all the histones and DNA are color-coded. What we see here, the interface, unlike the right offset stack, is made up of this H4 N-terminal tail only. And this is a zoomed view over here, and you can see the H4 from the bottom NCP, which is NCP1, goes upwards and interacts with the H2B of NCP2. Similarly, the H4 of NCP2 comes downwards and interacts with the H2B of NCP1. So these interactions were pretty interesting, but we wanted to know whether our structures are unique in any possible way. So as of September 2024, from the PDB, we downloaded all structures of the nucleosomes that had been reported by then. So that included 666 PDB structures. That does not include ours, by the way. And then out of all these structures, they were either deciphered by X-ray or cryo-EM. And by visually aligning them on our NCP structures, we identified there were 29 unique NCP stacks, and this 29 includes our 3 NCP stacks out of 4. And so in order to understand how the NCP-NCP stacking occurs, we only considered those NCP-NCP stacks where the disk of the histone faces the other disk of the histone. So what it means that they are parallel to each other, not arranged in the angle like 90 degrees like a L. Okay. Because some other papers reported previously have considered the L kind of arrangement as NCP-NCP stacks, but we have excluded that out of our analysis. So in order to understand the orientation of NCP2 with respect to NCP1, we performed a reference frame wherein we took the nucleosomes and used all the coordinates of the DNA double helix to identify the center of mass of the nucleosome. We removed the histones and just took the DNA coordinates to identify the center of mass. After that, we took we have the diet center over here, as we know, and 19 base pairs upstream and downstream of the diet was considered as the centroid. So centroid 3 and centroid 2, and then we could draw a plane including the diet, centroid 2, centroid 3, and center of mass. We could draw a plane, and that plane, when extended, could reach to the centroid of the 38th base pair upstream and downstream of the diet. And so now we can have a plane over here which slices the nucleosome. So imagine this plane being on the center for NCP2, NCP1 and NCP2. And when we have two planes, we can actually calculate how the NCP2 is oriented with respect to NCP1. So N1 is the nucleosome 1 here, nucleosome 2 over here. Now, another thing for you to remember is I have color coded the diet in green and the two sites, CO2 and CO3 centroids, in different shades of pink, light pink and a bit darker pink for the analysis. So the other thing that we can calculate was like how the NCP2 diet center is oriented, which is in green over here, which becomes CO5 now, with respect to NCP1, which is CO1. So basically, the diet center of NCP2 is turned in an angle to NCP1. And further, we can also calculate angular and rotational or angular and translational parameters. So the rotational parameters are the roll, twist, and tilt. And the translational parameters are the shift, slide, and rise. So here are some interesting cases out of the 29 in the essence of time. I've limited it to just 2 important cases. So this is a crystallographic obtained structure of a mononucleosome, and this NCP-NCP pack was determined by two-fold symmetry operation performance. And then there was a very nice kind of interface, which was mostly stabilized by the ion. What we saw here is the diet center of the NCP1 is completely 180 degrees rotated when we compare with NCP2. So like completely turned 180 degrees. However, this only happens in X-ray structures of mononucleosomes. In cryo-EM structures, normally they face in the same direction, the diet centers, except in the case of these telomeric nucleosomes, a nucleosome stack, which was determined a couple of years ago, where the researchers used one single DNA, and it it twisted in a left-hand orientation to form this nucleosome stacks, as you can see here. In this case only, you could see high shift values, which means here the NCP1 diet center is over here. The NCP2 is kind of turned, and the shift value is around 25 to 27 angstroms. And this is the largest shift value that we saw. But the NCP-NCP distance is the closest. And that is because there is no linker DNA between the two NCPs. And in our case, like I showed you before, the parallel stack and for the right offset stack, the diet center faced towards us, in one direction. And we have the beautiful interface that is stabilized by H4 in the parallel stacks and by H2B in the case of right offset stack. Now, taken together all the 29 structures, if we plot all the parameters here, I'm only showing the slide, that is the nucleosome distance versus the slide parameter. You can right away see that they have a characteristics V shape. What that means is that at higher NCP-NCP distance, the slide can be either positive, so the NCP2 can be moving towards the left, which is negative slide, or towards the right, which is the positive slide. But at lower NCP-NCP distance, that means when the two NCPs are closer, the slide value is close to 0, as you can see here. Now, where are our structures, for instance? So we have NCP parallel stack over here, which has very little negative slide, and then NCP-NCP distance is 63. And then we have NCP right offset stack, which has a little bit higher slide, as you can see visually as well, which is 30 angstroms, and the distance between the two NCP is 66 angstroms. Now, taken together, we'll just discuss this translational parameters, which is shift, slide, and rise. What we observed is that the slide value has a larger spread from negative axis to positive axis, positive values. However, the shift and the rise values were had a limited spread. So next we went to do the cryo-EM studies of the gamma H2X NCP plus BRCT complex. We prepared this complex in large scale and loaded on the size exclusion, and as expected, upon phosphorylation, the gamma H2X NCP and the BRCT come as one peak, and this is only for MCPH1, and the same is true for MDC1. However, when we have non-phosphorylated form shown in dotted lines, we had two, because the BRCT, which is a small protein, does not actually bind peaks to the NCP. And to remind you guys, this is the EM grids for the gamma H2X NCP, and you see stacks here. However, when we had BRCT in there, we could not see any stacks. Upon 2D classification, we just saw mononucleosomes, and we also saw the structure of it. However, we could not see any clue of BRCT whatsoever. So then we thought, like, what's going on here? So we performed molecular dynamic simulation. We took our crystal structure where we have this BRCT domain, and we have the phosphopeptide bound there. And after a 1 microsecond simulation, still the phosphopeptide was bound at the same place, and the terminal tyrosine was buried at the same place. The structure of the BRCT domain did not change. However, when we performed control simulation, that is, we removed a phosphoserine and placed a serine instead, so remove the phosphorylation. In no time, the serine popped out, as well as the tyrosine popped out. So basically, the peptide protein kind of moved away from each other. However, there was no structural change whatsoever in the protein. So the BRCT domain was intact. So now we got our trust on molecular dynamic simulation that, yeah, it actually mimics what we see in our bench, which is in test tubes, that without, in the absence of phosphorylation, the peptide actually doesn't bind to BRCT. So we thought to move ahead to do the same with the phosphorylated NCP instead of the peptide. So here we have the nucleosomes, the structure that we solved, and then we added the long tail to it with phosphorylation and also control without phosphorylation. And we performed one microsecond simulation, and what we saw is that the BRCT was bound to the tail. The phosphoserine and tyrosine was intactly bound. However, they were like flopping around, and with time they kind of settled in at different orientations on the nucleosome disc. And we also performed replicates, by the way. However, when we check the interactions, other interactions other than the phosphoserine and tyrosine interaction of the BRCT with the nucleosome, we actually could not find any specific interactions over there. So then we removed the phosphorylation. So basically, we had serine instead of phosphoserine, and quite quickly, the serine flipped out of the pocket as expected, and also the tyrosine actually moved out of the dislodging the tail from the BRCT, and that kind of confirmed once again that we can trust the simulation that the BRCT does not bind to H2X containing nucleosome. The phosphorylation is necessary. And additionally, the lack of phosphorylation on the serine also kind of eventually removes the tyrosine 142 from its hydrophobic binding groove. Now, we tried to think about this in another way. So we looked at the electrostatic potential surface map for the BRCTs. Here is for the BRCT of MCPH1, and this is for MDC1. Now, the phosphoserine and the tyrosine binding pockets are shown here, and they are positively charged in both the cases. But when we flip the other side of the BRCT, that is non-phosphoserine binding site, we do not see extensive positively charged patch either in MCPH1 or in MDC1. And why we need that positive patch is because, as you may know, that the nucleosome is highly negatively charged. One, because of the phosphate groups of the DNA, and two, because of the acidic patch. So in order for the BRCTs to sit on these nucleosomal surface, they need to have extensive positively charged patch to have charge-charge interaction. So now it kind of makes sense that yes, the BRCT actually only recognizes the phosphoserine and tyrosine, but not any other residues of histones or DNA. So in other words, the histone, the nucleosome structure remains intact. Now, we also went back and looked at the literature and did a little bit of structural analysis. And literature says that after the BRCT of MDC1 recognizes the H2AX nucleosome, it recruits using its N-terminal FHA domain, it recruits RNF8, and eventually RNF168 is also recruited to the nucleosomes for ubiquitination. Now, if you look at the structure of the nucleosome disc, and now this nucleosome disc is kind of like in a horizontal manner, and here you see the H1, which locks the DNA at the entry-exit site. And here you have shown in green is our histones, and this is the H2AX tail. And you have the phosphoserine here and tyrosine buried here, and this is our BRCT model. Now, the N-terminal of the MDC1 is the FHA domain that recruits the RNF8, which mediates the ubiquitination of H1. Now, this location of the H1 and the H2AX tail are quite close to each other. So basically, the that also suggests that the BRCT cannot actually sit in this region, neither on the nucleosome or on the DNA. Now, what about the other side? Now, the RNF 168, and there is a cryo-EM structure that has been solved, where the nucleosome is bound to RNF 168, and it shows that it binds to the diagonally opposite face of the diet center, so which is over here. That means the BRCT is also not able to, does not have any charge-based interaction with the negatively charged acidic patch, also will not hinder the binding of RNF 168 because this is its business end. So now, another way to think about it from our structure is here. The C-terminal tail of H2AX could be built till lysine 118. Now, we have this long stretch of residues, like 22 stretch of residues till we reach serine 139. And now, as you may know, the C-alpha between neighboring amino acids is 3.8 angstroms long. So in an extended conformation, this stretch from 118 to 139 will be almost 83 angstroms. Now, from our experimental cryo-EM structure, we already have noticed that the center of mass distance between the NCP1 and NCP2 is only 63 angstroms. So how can it accommodate such a long stretch if it is in extended conformation, this tail bound to the BRCT? Also remember that the diameter of a double-stranded DNA is 20 angstroms. So taken together, what we see is there is steric hindrance if we want to pack this BRCT within a stacked nucleosome structure. And no wonder this explains why having the BRCT bound system actually disrupted the stacking. So in summary, we purified H2AX containing nucleosome and also gamma H2AX containing nucleosome. We also purified homogeneous preparation of BRCT gamma H2AX NCP complex. When we applied it on grids for the nucleosome only particles, we did see interesting stack structures which has not been reported before. The stack-to-stack interactions, the NCP-NCP distance in the stack interaction was almost 67 angstroms, like within that range. And the different kind of stacks that we saw, parallel stack, right offset stack, left offset stack, they kind of mimic how the nucleosomes may be packed in a heterochromatin organization in the nucleus. Now, for the BRCT bound system, we did not see any stacks. We prepared the model and performed molecular dynamic simulation, and what our simulation suggested, including our negative controls, suggested that this packing has to be disrupted for the BRCT to recognize the phosphorylated gamma H2AX tail, and thereby leading to the recruitment of DNA repair factors downstream. And so with this, I would like to highlight that these cryo-EM, single particle cryo-EM structures reported from our lab are the first set of structures that were done, like from the project design to the structure solution was all done in-house in the absence of EM facility at U of A. And so I'm very grateful that I was included in this team by my supervisor, Mark Glover. And thanks to his vision, his persistence, and that we could do this project. And thanks to the lab members, and particularly all these people who worked with me in this project. And like I said, we didn't have a microscope, so we used to prepare grids and collect data at University of British Columbia, UBC, and the Stanford microscopy facility, and we solved the structures in our lab. With that, I would like to thank all the funding agencies and also Memorial University to help me set up my own lab. And I'm looking for students now. And this is my email ID for future contacts. Thank you all, and I will wait for your questions.
Thank you, Rashmi, for your interesting talk. And we can now welcome questions for Rashmi. If you have a question, please type it in the question box as shown on the image on the screen. So there is already one question about NMR. I am not an expert in nuclear magnetic resonance, and the question asked is, why do you think that when you search for unique NCP stacks, none of those 660 plus PDB structures were obtained with NMR?
I'm not an NMR expert, but with very little knowledge, I think the NMR experts in the audience can chime in. But I think the NMR, this oligomer is quite large in size, I think. But then again, I'm not an expert, so I think it's because of the size.
Okay, thank you. I have a very general question for you while we get more questions. Please ask us more questions. So, you know, not all nucleosomes contain the histone variant, right? And so you describe this key interaction with BRCT containing proteins. And so how, you know, your experiments are done in vitro, how do you think in vivo the content of the histone variant will affect chromatin compaction and larger levels of chromatin organization?
Yeah, so that's a very interesting question, and I have thought about it throughout my work, because there is no like actual literature talking about it. So this is just my imagination. And so, as you know about the phase separation concept, right? And so in the nucleus, we have this chromatin compartments, which is like open chromatin and then closed chromatin compartment, instead of the traditional accept chromosome, which is now that theory is not that strong, right? Strongly valid, but because of that fluidity of the chromatin, it can happen that the nucleosomes which contain the H2X variant, which actually is like, I think, less than 15% in the cell, those nucleosomes from far can come near to the DNA repair site, where they get phosphorylated, and then they can trigger the DNA damage signaling response. That's my imagination.
Okay, thank you. Sergi Consolato asks, there are advantages and disadvantages linked to microscale thermophoresis. However, one major disadvantage of label-free thermophoresis is that protein-protein interactions cannot be measured, as generally most proteins have enough aromatic residues to generate an interfering signal. Can you please give more details of your experience with microscale thermophoresis?
Yeah, thank you for the question. And it's you're right. Actually, in our case, to avoid that, we used a labeled system actually. So it was not label free. So the BRCT's histidine tag of the BRCT was labeled, and that avoided that kind of biasness actually.
Okay, thank you, Rashmi. Mariam Ishkaya, what roles do histone modifications play in altering nucleosome mobility or accessibility?
Yeah, like I mentioned in probably my third or fourth slide, the histone tail modifications, they're very specific. There are 60 different kind of modifications known till date, but they're highly specific and highly regulated. For the process to start, they are installed, and after their installation, they either recruit different binding factors or by themselves cause changes in the chromatin structure, condensation or decondensation to trigger the process. Does that answer your question?
Thank you. Tisin Rubab asks, what are the future prospects and projects in your lab? What are you doing next?
So we are advancing towards the ubiquitinating machinery, like trying to more understand in my lab the ubiquitinating machinery, how it works in context of the nucleosome, because after the phosphorylation, it's the next step to understand is the ubiquitination, and there is a lot more to learn there. Although amazing structures have been reported, like cryo-EM structures of the ubiquitinating machinery, but yeah, it's challenging. So.
Can you tell us anything in advance of how it affects?
So as of now, of course, the stacks will not be formed anymore because of these polyubiquitin chains. But as of now, because I'm just starting the lab, so maybe in a year I could present.
Good. What do you think contributes to the difference you see between cryo and X-ray structures?
Yeah, that's a fantastic question. Actually, the X-ray structures are more of mononucleosomes, but the packing is due to the crystal packing. And when the mononucleosomes have been reported, the ones that are crystallized have been reported, the second NCP, the NCP2, is most of the time like kind of oppositely oriented, that the entry exit site are completely diagonally opposite to each other. And so I am not sure if this is biologically relevant, but this is how the crystal packing works, and they're generally stabilized by ions, right? So it's like charge-based interactions and pretty nice fit, and also high resolution structures. However, in cryo-EM, it is personally, maybe I'm biased, but more natural, let me put it that way. There are two kinds of cryo-EM structure, if I may say, excluding ours, if I may say. One, cryo-EM structure of stacks which have long linkers. And when they have long linkers, the entry-exit site, they face towards in the same directions for each stacks. And then the second one is are the ones I showed you, which has no linkers. In that, all the nucleosomes are tightly packed, so there is no space for the H1 histone. So they are just tightly packed, like in the telomeric nucleosome stacks. Beautiful structures, but because of the tight packing, they don't slide. There is just little bit change in orientation towards the positive side for the diad axis, which is the change in shift, but there is no slide. They are like tightly packed. So yeah, and if I may say, as of now, ours is the highest resolution in terms of cryo-EM structures. So that's
Okay.
Yeah, but I'm sure in a couple of months there will be more structures which will be much better resolution.
One more question, maybe we have time for it. So from Fruit Tower 3K27 mutations would impact wrapping, nucleosome wrapping or unwrapping. How do you think? Yeah.
So, I mean, I would like to predict, but unfortunately, like what I did with this project, we could not predict anything, right? Everything was unpredictable because we wanted to see the BRCT bound to the phosphorylated tail bound on the nucleosome. That is the structure we expected, but what happened is completely different. And we did experiments after experiments, both in silico, went back to theory and studied more, and then made a conclusion, and then figured out that, okay, the BRCT can never bind to the nucleosome actually on the disk except the phosphorylation. Similarly, I think in your case as well, for the K27 mutation, personally, I would find it hard to like predict something. I think it's the best way is to do and check what happens.
Okay.
Because the magic underlies the chromatin dynamics.
Yeah, and there is lack of resolution. So we need to thank Rashmi for her interesting talk and for taking the time to answer all the questions we received. Thank you everyone for attending and the speaker. And you can continue the conversation online if you follow Biochemical Society on Blue Sky and X. We welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. And if you have an idea for a webinar in 2026, we invite you to submit a proposal for it. And you can find more information about webinars at biochemistry.org. All our webinars are listed on the website as well. And if you have missed any of the 100+ webinars that we have already run as part of the series, or would like to watch them again, please visit our website or YouTube channel. The recording from today's webinar will also be available within a couple of weeks. And finally, I want to highlight that it is more important than ever to stay connected and engaged with our fellow molecular bioscientists. So please join our Biochemical Society community of researchers and specialists and take advantage of all the benefits, including discounted registration fees for society courses and meetings, and exclusive access to a wide range of grants and bursaries, or personal online access to 2 of their journals. So visit more on our website. And thank you all, and we look forward to see you again.
Thank you, Lucas. Thank you, everybody.